So hi, everybody. Thanks for joining. This is our SB Grid. Well, it's been our weekly seminar series since the start of the pandemic. This is our final one of 2021. We're going to take a break for the summer. It is definitely summer here in Boston. I think it's almost 100 degrees here today. I'm not exaggerating. It's like 199 Fahrenheit or something. So summer is definitely here. So I'm going to take a break and kick it back off in the fall. It is my pleasure to welcome Alexandre Bonvin from Utrecht. So, and Alexandre's going to tell us about HADDOCK.
Okay.
So, and with that, go right ahead, Alexandre.
Okay, thank you very much. It's a pleasure to speak again in this SB Grid webinar series. I guess everything is fine. You're hearing me and you're seeing my slides. So I'm going to talk about HADDOCK, or software for integrative modeling of biomolecular complexes. So I'm Alexandre Bonvin, based at Utrecht University. And this is Utrecht here on the map of the Netherlands. And if you don't know where the Netherlands is, then you should look it up. Utrecht, if you ever come into the country, is definitely a place you should come visit. We have this beautiful center, an old city full of terraces, which are now also alive actually after a long period of lockdown. So it's really an enjoyable place. Our university, this is the academy building in the center of the city, which is an old building. The university dates of 1636. And we have, of course, a new campus. And our university is very proud these days to have, since less than 2 weeks, I think, the longest rainbow bike path of the world. And on the campus, you will find this building. And in this building, this is where my office is. But today I'm speaking to you from home, since we are still have limited access to the facilities. So this is the topic for the next, well, about 45 minutes. I'm going to put everything into more general context. Then I will speak about the specific of HADDOCK as a software, describe some of the very versatile way of defining restraints to guide the modeling process, show you some of the more recent developments. The last talk I gave was, I think, in 2017, or if not before, and I was speaking about HADDOCK version 2.2. Now we are up to 2.4, and I will end up with some general information about software, its use, and some practical tips. So where are we in terms of software? Where would we stand? We stand in the field of structural biology of interactions, where on the right side you see here experimental methods, the classical method to study structure, mainly X-ray, cryo-EM, and NMR, allowing you to get to, say, atomistic resolution, but also a lot of other experimental methods that are providing you pieces of the puzzle. And as you move to more complex and maybe dynamical assemblies, you will have to use a variety of methods to get to the final product, which will be a description of those systems at an atomistic level, and ideally both structure and dynamics at the same time, which is not an easy thing to do. Which is why you also need, next to the experimental methods, you need computational methods, which brings us into the world of computation. And there are different methods to generate structures or model of biomolecular assemblies. You can do it by homology modeling or threading, provided you have enough structure in a protein data bank of other complexes that you can build onto. But for today, the context of or computational modeling will be docking. One of the last webinar, if you attended it, was about LiDock, so there was a lot of information about docking in general. So this is docking in a nutshell, giving the structure of two biomolecules, protein in this particular example. Can we predict how those interact? Can we predict a 3D model, generate a 3D model of their complexes, which requires sampling space, possibly trying all translation and rotation of those molecules, and measuring the quality of the model based on a number of geometric or energetic terms or statistical terms, machine learning even these days, where the shape complementarity will play a role, but also energetic terms like electrostatic energy, you see a Coulomb potential, and van der Waals interactions, you see a Lennard-Jones potential. So this is the game we are in. Now, so you have this interaction landscape, so interaction space between the molecule, and everything here is represented as a two-body system, so two molecules, but many assemblies consist of more than two molecules. So you need also to be able to go to larger assemblies and larger number of molecules to deal with, which increases, of course, greatly the complexity of the problem. But so we have here on the x-axis this interaction space, and we have some kind of functions that measure the quality of a model. And the game is to sample this energy landscape with as hope to find the global minimum of this landscape, assuming that our energy function points to the right native-like conformation of those complexes. So sampling is generating the models, and then we have the scoring problem in docking, which is basically assigning some score to all the models that we generated with the aim of distinguishing what should be the global minimum. Now, if you have data, you can choose to use the data in different ways. So you can choose to use the data to bias the sampling. This is what we are doing in HADDOCK. Or you could use the data only to do scoring. And there are other software. There's a lot of docking software that are doing basically full sampling of the interaction space, so ab initio docking, and then use data to basically identify the putative native complexes. And in HADDOCK, we are actually doing both. So we're using the data during the sampling to bias the search, but also using the data as part of our energy function to score the models. Now, what kind of data are we speaking about? Which brings us to the field of interactive modeling. So there's a lot of experiments out there that are giving us, as I said before, pieces of the puzzle. So those pieces might be simply the knowledge that one amino acid is very important for the interaction. So you can get that from mutagenesis. You might get that from NMR titrations, where you map the interfaces without knowing how things are binding. HD exchange detected by NMR or mass spectrometry these days. Protection from exchange might point to possible interfaces, but also a lot of experiments that are going to give you some kind of distance information. Classical example, cross-linking detected by mass spectrometry will provide you some distances between two amino acids, and the distance will depend on the chemistry which is used in the linker. But think of FRET experiments, DIR experiments, so any type of experimental data that can be translated into a distance in principle can be used in HADDOCK. You might have orientational information. This is data coming from NMR. And you might have also shape type information. So cryo-EM, small angle scattering, neutron or X-ray scattering, cryo-EM if you don't reach the atomistic resolution, and there are still plenty of cases. Cryo-electron tomography will probably be still have a hard time to get to atomistic resolution where you could build your complexes from scratch. So there, if the resolution is more limited, say medium 5 to and higher up, you might need to resort to docking method, modeling methods to build the complex into those shapes. And this is again information which in principle you can combine into some kind of software, integrative modeling software, to get the answer that you are looking for. So this was the general introduction in terms of where are we sitting in terms of field. And now we're going to move more into the specific of HADDOCK. So HADDOCK is now almost 20 years old. The original publication was in 2003, and the idea, of course, came before that. The idea came from NMR data, where we were getting those titration experiments where these are pointing to where things are binding, or which region of the protein is affected by the binding, but it's not telling you how it's binding. And this, at the time, we figure out a way of basically using this information in some kind of restraints to guide the modeling process. And I already mentioned all those information sources, but these are all basically data sources which you can plug in one way or another into HADDOCK to guide the modeling process. And if you don't have experimental data, of course, there's the entire field of bioinformatics, coevolution these days, where you can extract distance restraints, but also prediction of interfaces, which can be used again to drive the modeling process. Well, since the early days of HADDOCK, we have extended the capabilities to the modeling of up to 20 molecules. At the same time, symmetry can be leveraged also in terms of restraints. As you will see, we do allow for flexibility at the interface between molecules, which is important because biomolecules are not rigid, they are not stone, but the flexibility is an interesting part of the recognition process of their function. So how do we encode information which you might have on your system telling you that some part of the protein or some surface of the protein is important for the interaction without knowing what are the exact contacts that are made. And this brings me to the concept of ambiguous interaction restraints. So ambiguous distance restraints. So it's not a restraint which is now defined between 2 points, but it's a distance which is defined between a collection of points. And the concept was introduced in the early 90s by Michael Nigius and Axel Brunger to deal with ambiguity in NMR data. So basically, you calculate you see here a classical function which is used in NMR structure determination, and this is also the function that we use actually in HADDOCK. So you have a view of this function here. So there's a region, there's a lower limit to your distance, and there's an upper limit. If your distance is in between this range, the energy will be 0, meaning that there is no force acting on a system. If you are below the lower distance limit, then you have an harmonic function going up. And if you have above this function, you have again an harmonic function, but it's harmonic only for a short distance, which can be tuned. So if you are between this U and S part, you have the harmonic function, the classical potential. But as you move away from that, longer distances or longer violation, your experimental data tell you should be somewhere in there, but your model is somewhere in here. So we are switching from harmonic to basically a linear function. So the energy still goes up, but the advantage here is that the force becomes a constant. And if the force is constant, then your simulations are more stable if you use force in your simulation. And in HADDOCK, we use gradient-driven optimization techniques like energy minimization, molecular dynamics, where the force are basically driving the optimization. Now, this is just the energy function that we use to encode the information, to encode any information that can be translated into a distance restraint. So now what about the distance that enters here? Because U and L are basically experimental bounds that define what your distance should be, but you need to input a distance in this function R. And the distance that we input in this function is this effective distance, which is calculated as the sum of all possible pairwise distances between a residue which is defined as being part of the interface in your first molecule and all residues that are defined as being part of the interface in the second molecule. So we do this sum of 1 over the distance to the 6th power. So 1 over r to the 6, if you are an NMR expert in the audience, you recognize this as being dipole-dipole interactions, which is the reason actually why this was introduced, because the first reason to use this kind of averaging was coming from NMR. If you are now a more modeler, you will also recognize that this is the attractive part of Lennard-Jones potential. So so you take the sum of all individual distances between the interface that you have defined, and you have one such distance for each residue that you define as being important for the interaction. And when you have summed all those distances as 1 over r to the 6, you take the inverse 6th root of that. And that gives you one distance which enters this energy function. Now, the property of that distance is that it's always shorter than the shortest distance that enters the sum. Meaning that it doesn't matter which contact are being made. If there is a rather short distance in the plethora of individual distance that you calculate, you are most likely to be in the region of your energy function, which will be 0. So you will be satisfying your distance restraints. Now you define such a distance restraints for each of the residue that you identify or predict as being part of the interface. So you have a network of distances between those interfaces. And those basically effectively pull the molecule together without predefining in which orientation the complex should be formed. So that's the ambiguity level. And depending on the number of residues that you define here as being important for the interaction, which we call active residues in HADDOCK terms, you might have up to thousands of those distances that are summed up. Now, since typically the data are also never perfect, what we do by default in HADDOCK is to randomly delete 50% of those distance restraints for each model that we generate. And this can be increased to up to 90% if we don't trust the data very much, or if you, with bioinformatic predictions, for example, we like to over-predict to make sure that you cover the interface, which means that you have a lot of false positive predictions in your data. So there we might remove a higher percentage. Of course, if you have a trust in your data, you don't need to remove those. Now, next to this distance restraining energy term, and we have other terms that represent our type of data, but next to this term, we have our classical force field, which describe bonds between atoms, angles between free atoms, rotation around bonds, torsional potentials, and non-bonded interactions. These are important for molecular recognition, of course. So we have van der Waals interactions and electrostatic. And here we use energy functions that are like Coulomb potential, Lennard-Jones potential. So these are the similar functions that are used in molecular dynamics simulations. So these are not the functions that might be used when you do classical structure determination by NMR or X-ray. X-ray, often you don't use electrostatic. Here, for the modeling process, electrostatic is very important. And we're going to try to minimize this energy function using a combination of energy minimization and molecular dynamics simulations. So the protocol in HADDOCK consists of 3 stages. In the initial stage, we're going to treat the molecule as rock solid. So it's a rigid body energy minimization process. Then we are going to refine the interface using some kind of simulated annealing scheme. So we start at high temperature and cool down the system. And at the end, we might solvate the molecules into water and then do a very short refinement of the models. So this is how this rigid body minimization looks like. What you see here is one of the first complex calculated with HADDOCK, where the spheres indicate residues that were identified by NMR titration experiments. And those are basically driving the interface together. So compared to ab initio docking, we are really targeting the interface directly and minimizing this interaction. Now, this process starts from random orientation of the molecule. So you have to repeat this many times to sample enough interaction space. This is quite cheap. So we typically sample in the order of 10 to 100,000 solutions at that time. And we might write to disk in the order of 1,000 to 10,000 models, single structures. So once we have done that, we are going to do a first selection of models, and we select a fraction of those models, 10, 20%. And then we go into the refinement stage, where we now introduce flexibility into the system. So this is rigid body dynamics. Now you see that the side chains are moving. So we optimize first the interface at the level of side chains, and then we add flexibility in the backbone. You just saw loop switching here, flipping over. This is happening in torsion angle space. The computational engine, which is used by HADDOCK, is actually CNS, crystallography and NMR system, which is related to Explore, which is related to CHAR. So this is the computational engine where we do actually the effective calculation, and HADDOCK itself is a Python framework, which is sitting and harmonizing everything and making sure that the entire workflow works. Now, at the end, we might solvate the molecule into a layer of water and do a very short molecular dynamics simulation. Don't expect miracles in terms of conformational changes in those different stages. How much conformational changes you might be able to induce will also depend on the amount of data that you have at hand to guide the modeling process. So if you have no data, then you should not expect very large conformational changes. So changes of up to 1 to 2 angstrom maybe are doable. We have seen changes, conformational changes of maybe up to 5 angstrom in some cases where you have really good data. But this depends very much on your system. So we are not going to fold the protein onto the interface of another protein. This is simply not doable. So flexibility is a big challenge in the modeling of interactions. So what are the treatment of flexibility, which means that we can provide an ensemble of conformation to start the docking process. So you might want to pre-sample conformational changes prior to the docking. For example, if you do antibody modeling, you know that H3 loop is one of the challenging ones. By pre-sampling the conformation of H3, maybe one of the conformation that you generate might be better suited for the docking. And we let HADDOCK basically select which one of those at the end works better. And then we have the explicit treatment. We basically allow explicitly side chain to move and the backbone to move. So what about the energetics in the entire process? So we use OPLS parameters. So the force field, which is actually an old version of OPLS, United Atom version, rather short cutoff for those of you doing molecular dynamics, but this was kind of optimized to get reasonable results in a shorter time. And important at the end, we cluster the solution that we are getting so that you don't look at, say, hundreds of models, but you look at a few, maybe less than 10 or a few tens clusters at the end. Now, how do we score the different models? So we have different scoring function at the different stage of the modeling process, because here at the rigid stage, we might sample 10 to 100,000. And at those stages, we sample a few hundred typically. Now, the final score is a very simple function, which consists of the van der Waals intermolecular energy between the molecule, 20% of the electrostatic, a dissolvation energy term, which accounts for the basically removal of water from hydrophobic surfaces, which is usually a bonus in energy. But if you dissolvate a charge residue, usually you pay a price. And then we have the experimental information, which also adds up to the score. So the function that was published in 2003, it's still the default function in 2021. We have been optimizing it, and of course you can optimize it for very specific systems. But at the end, this is robust. So it does the job for a variety of systems, and even if you start mixing molecules, which doesn't mean it cannot be better. So most of our users are actually using the web portal. HADDOCK is also installed, of course, in SBGrid, and the 2.4 version is there. So we have more than 23,000 registered users and served a very large number of docking runs since the server went online in 2008. And we are able to offer those resources because we have access to distributed high throughput compute resources, the so-called grid resources across Europe, but also Asia and the US. For example, HADDOCK is sending job to the Open Science Grid in the US. I'm going to come back to the server later. So this is our world map of users, which I made this today. So we have 23, and you see for the US participant here in the audience, you are the third. So EU aggregated is slightly higher, and then India has a very large number of users, and this is fluctuating from time to time. We also have seen actually over last year, speaking of COVID, that people have been, probably when they were in lockdown, they could not do experiments. They started using, discovering computational resource much more. And what you see here is the usage statistics over the last 2 years, since January 2019, actually, more than 2 years. And we have quite a constant usage, about 3,000 docking runs per month being processed by the server. And here comes the lockdown, and we have almost a tripling of the usage. And we seem to see the COVID waves as well in our usage of the portal, which might be just pure hazard or pure luck, but it's interesting to see. What you also see in orange is that we start tagging, asking users to tag their job as being COVID related, so that we can also report the usage for COVID related research, since we rely on grants, of course, and funding agencies to do all this work. So it's good that we can also show why, what it is used for, especially during this pandemic period. And not only the number of jobs has more than doubled, we also see that the number of single active users per month has also doubled. So we had about 300 users per month who were active, and now we had a peak at more than 600, and actually in the second wave, we are reaching almost 800. Yeah, so this was something that came out on the internet. So by the way, if you think, why did you call your software after a fish? HADDOCK, of course, it's the name of the fish, which is nice because there is no copyright associated with the name. But HADDOCK is also the captain, who is the big friend of, Tintin. And you don't see them very well here because they are all geared up to deal with the COVID pandemic. But actually, this picture here is a combination of Tintin going to the moon. That's what they are wearing here, and the Blue Lotus, which is happening in China. So next to HADDOCK, just a side step, our lab is developing a number of other software, some of which are also actually on SPGrid, like PDB tools, this, I think in Prof as well, if I'm correct. But these are also available as web portal from this address, winmrscience.nl. So now that you have an overview of HADDOCK and what it can do, let's dive a little bit more in and to show you how versatile HADDOCK is in terms of defining restraints between molecules. So I already explained this concept of ambiguous distance restraints, but restraints, of course, don't have to be ambiguous. If you have very specific information, you can input it as very specific information. But you can input things in HADDOCK in 3 different categories. So it's called ambiguous restraints, unambiguous, and hydrogen bond restraints. The name doesn't mean that you have to put per se ambiguous restraints in the ambiguous category. We should maybe have called them apple, bananas, and oranges. But there is a different behaviors of different type of restraints. So everything that you put here in the ambiguous category, by default, 50% will be randomly removed. So if you don't trust your data, you might classify them as ambiguous. If you trust your data and you want them to be used all the time, you could put them in this unambiguous category. And hydrogen bond restraint is something we added to sometimes maintain secondary structure. And by default, they are not used in the initial docking stage. So they will not guide the docking, but they only use in refinement stages. Now, how do you define these distance restraints? Especially if you use the local version of HADDOCK, you will have to generate those distance restraints yourself. And the syntax is based on CNS, the software, the computational engine behind HADDOCK, and Explore. So if you know CNS Explore syntax, then it should be very natural to you to define restraints. Here are a few examples of such restraints. So you see here, so this will be an ambiguous restraint that defines that residue 38 on chain A. So you see there is no atom definition, it's the one residue. So this could be a residue defined by mutagenesis. So you have a distance is defined by two selections. So you have a first selection, you see two brackets here, and the second selection is much longer. You open the bracket here and you close it here. And here there's a list of residues, and all those residues will be basically the residue that you have identified or predict as being part of the interface in the second chain, chain B. So this can be a long list. And you see here three numbers. So this is the target distance. This is the lower bound correction. So this minus this gives you the lower limit to the distance, which is 0 in this case. And this plus the third number gives you the upper limit, which is 2. So we use an upper limit of 2 angstrom to define distance between proteins, which might seem very short because, you know, minimum approach distance of 2 carbon atom is around 3.5, say 3 angstrom. So this is shorter than the minimum distance between 2 carbon. But because of this averaging function, the sum of 1 over the distance to the 6, the effective distance that comes out of the sun is always shorter than the shortest distance that you put in. And that's the reason why we use this very short distance. Now here you have an example of a restraint which is extracted from mass spectrometry cross-linking data. The range is 0 to 23 angstrom, and this upper limit of 23 angstrom is defined by the chemistry of the cross-linker that you used. And you see now the distance is specified to a specific atom. We apply it to the C alpha carbon in that case, between 2 specific residues. So there is no ambiguity in this distance. And the range is 23 minus 23, 0, to an upper limit of 23 plus 0, which is 23. And here you have a more specific distance restraint. So this is one that we used for protein nucleic acid interactions. And you see that, so this is a residue of the protein, no specific atom defined, but the specificity in protein nucleic acid recognition must come from the bases and not from the ribose unit. So we specify the restraints to atoms that belong to the base of the DNA nucleic acids, basically. So that's the way you could do that. But the syntax is extremely powerful, and then you can have a lot of flexibility in how you define those restraints. If you dive more into the syntax, selection syntax in Explore CNS. So what should you do? What should you not do? Some people have no idea where the binding regions are, and what they do is to define all residues of the protein as active. So this is generating effectively a n-squared problem because you're going to calculate all possible distances combination between all atoms in your 2 proteins, which is a pure waste of CPU resources. So you should not do that. If you have no information, either use the ab initio options of HADDOCK. So there is 2 types of restraints that were implemented, center of mass restraints of random patches that are selected on the surface and brought together, or use another ab initio docking software. Also, I already told you that you should not expect large conformational changes in this kind of modeling. So if your residues are buried inside your protein, it doesn't make much sense to define them as restraint. You want to restrain to solvent accessible surface residue. Okay. If you only have information on one of the two partners, typical example could be an antibody-antigen complex, where on the antibody side, you know where the loops are, so the hypervariable loops, but on the antigen, you might not know where the binding site is. You could, in principle, define the entire surface as passive. I didn't explain the concept of active and passive. Passive residues in HADDOCK, they can be at the interface, but they don't have to, while an active residue should be at the interface, otherwise an energy penalty is raised. What is very important is that if you go to more than 2 molecules, so you start doing multi-body docking, and the current limit in HADDOCK is 20, you should have good information and ideally specific interaction of which residue, of which interface interacts with which other partner. If you have very little information, this is going to be a waste of computational time to try to dock these very large systems. So what are other types of restraints supported by the software? So actually, my lab is based in an experimental settings within Utrecht University, the Bijvoet Center, and we are in the middle of an NMR infrastructure. So some of the first restraints that we built it into HADDOCK were actually NMR-based restraints. So from NMR, you can get information about the relative orientations of molecule, and this is something that might be very useful, especially in the context of interactions. So you have residual dipolar couplings, pseudo-contact shift, anisotropy restraints. The pseudo-contact shift are interesting because they give you not only orientation, but also distance information in one. When you deal with that, you need to have to define tensor, so it's a bit more complex, but it can be done. We can also define dihedral angle restraints. So this is affecting rotations around bonds. Of course, this has no effect on the complexes because you can only define those for within one molecule. We also have an option for radius of gyration restraint, which was added to kind of as a way of dealing with a bit with Sachs data. So it can be used, but in our hands, it doesn't do much. So we have other ways that we are working on now to incorporate Sachs data into the modeling process. I mentioned symmetry. Symmetry is also important. It limits very much the interaction space that you have to sample. If you know that you are dealing with a symmetrical system, the problem when you do docking is that you don't know where the symmetry operation should be, where your symmetry axis should be. So the way to deal with symmetry is something that was invented for NMR, for structure calculation of symmetrical dimers in NMR by Michael Nilges again. So we use the concept of symmetrical distances. So we define pairs of distances, and you see this is like a symmetrical system. You will see the amino acids, or say these are the C alpha atoms along the backbone. Distance b to x prime is symmetrical to the distance b prime to x. And if you define an energy function that requires that the difference between those symmetrical distance should be 0, and you define such distance along the entire backbone, you effectively enforce that your solutions at the end satisfy a C2 symmetry. And you can expand this ID to more symmetries. So currently we have a C2 to C6 symmetries that are available in HADDOCK. So this will define basically symmetry operation between your molecule, and next to that, as is done in X-ray refinement, for example, you might also define what is called non-crystallographic symmetry restraints. And this is effectively a restraint that say that if you were to fit the two molecules on top of each other, the root mean square deviation should be 0. So this does not define symmetry operation, but it makes sure that the conformation of your molecule are exactly the same. So both are supported in HADDOCK. Now, some more recent development now. So we've been compared to HADDOCK 2.2, which was the previous versions, we have been extending our capability of modeling up to 20 molecules. Disclaimer here, you should only do that if you have good information to guide the modeling process, otherwise you are wasting computational resources. Well, the details of this story was published in this paper, 2017. I also mentioned EM data, so we have built a way in HADDOCK to basically use the EM density. So we basically translating the EM problem into a X-ray crystallographic refinement problem. Since we are using CNS, which has all the X-ray energy functions, we can directly use the energy functions that are present too. And what we had to do is develop the protocol to be able to do that. This protocol makes use of centroids, so you have to identify first possible location of the molecule in the density. There are ways to do that. We developed our PowerFit software to actually identify those locations. And we first use distance restraints to pull the molecule into the density. And once the molecule are inside the density, we turn on the X-ray term representing the density, and we refine the complex. And this is compatible with all the other terms. If you were to turn on the density, EM density directly from the start, when the molecules are randomly separated in space, the system is not converging. So the reason to use the centroid is for conversion purposes. And this is now also supported in the web portal. And if you want to read all the detail, I'll point you to this structure paper. In order to be able to move to larger assemblies and model a more complex system, larger system with more than a larger number of subunits, we also implemented the Martini force fields into HADDOCK. So the current implementation has Martini 2.2. Martini 3 has just been released this year. So we'll have at some point to try to update things. It's implemented both for proteins and nucleic acids. And what's happening is that we are basically transforming our atomistic model into a coarse-grained model. And the Martini coarse-grained model has a 4-to-1 mapping. So four heavy atoms are transformed into one bead. So you have a smaller number of particles that you have to deal with, meaning that your computations are going to be faster. But this also has the effect that you are smoothing your energy landscape that you are searching for. So the docking then happens with the restraints that we have. There is no support for EM, by the way, at the coarse-grained level. But the docking happens at the coarse-grained level. And the final stage of the protocol is to basically to rebuild the atomistic model. So what HADDOCK will give you at the end is still a fully atomistic model. If you run a local version of HADDOCK, you will have to do this step, the generation of the Martini beads manually. There are scripts to do that, so we are providing a script. If you run at the server, then this is done automatically by simply switching one switch on the server. If you want to read about it, check those papers. This one is about the protein implementation, and this one is about the nucleic acid implementation. One example of using HADDOCK, if you want to learn more about what's the benefit of really using data to guide the modeling process, so this is an example we published last year, is basically to use information in this case from the knowledge of hypervariable loops in antibodies to guide the modeling of the protein of the antibody antigen complexes, and we compare the performance with other docking software that also have some options to use the data to guide or score the models. So I'm referring you to this story if you want to read all details. And something which is just out in bioRxiv a few weeks now is a way that is a new type of restraints basically that we have implemented now, where we use shapes to basically guide the modeling. The same idea is actually also being implemented to use SAX data. So by transforming SAX profile into a 3D shape model, bead model, and then defining restraints to place the protein into that model. So that might also be an option to model low resolution EM data. But what you see here, so this story is about modeling, it's small molecule docking, basically, and it's kind of a template-based modeling approach, where we identify a template which has a ligand, the ligand the most similar to the ligand that you want to dock, and usually there's a lot of information that you can find in the PDB. We then transform these template ligands into a shape with artificial bead atoms, and then we use distance restraints between the ligand that you want to dock and those bead atoms. This shape is not interacting from an energetic point of view with the remaining of the system, but there are distance restraints defined from your ligand to the shape to make sure that the ligand should be overlapping with this shape at the end. And we have two ways of doing that. One way where we only use basically the 3D shape information to guide the modeling, and another way where we define a pharmacophore model. So now we are associating properties to the different beads that we defined, which might be polarity, charge, things like that. And then the distance restraints become much more custom, so that you say that, for example, an oxygen atom in my ligand should be at proximity of a bead which is electronegatively charged. So you can read the story already on bioRxiv. Now, a few more information, general information about using the software and some practical tips. So where can you find the software? On SBGrid, then you don't need to install it. Everything is there. You can start using it if you want to use the local version. If you want to use make your own installation, then you should visit bonvinlab.org/software, where you find a link to HADDOCK 2.4, and there you can basically fill a form, and then we will send you the software. So registration is required, but it's free for non-profit usage. So this is what you see in this page. So you will find a link to the manual, which is a good starting point if you start going to use the local version of the software. There is also a link to the web portal. You get this license form, basically the form that you need to fill, and you also find a link to our support center. I'm going to come back. And very important, if you have never done any haddocking in the past, it's a good idea to start with this best practice guide. I'm going to come back. You will not only find HADDOCK in this software site, but you also find other software which are being developed in a group, like PDB tools I mentioned, collection of Python scripts to manipulate PDB structure, which is available also on SBGrid. I mentioned Parfit to identify, for example, centroids in the densities there, this, so have a look of what's there. The manual, also a good place to have a look if you're going to start using the software. So you have some general introduction about how things are happening, a lot of information about defining restraints, and you will also find in there information about the format of those restraints, if you're going to generate those, different parameters related to the docking, how to set up the docking, what is the scoring function used per HADDOCK, analysis, if you want to do a manual analysis, which you would have to do if you run a local version, so how to do that, and also some tips and tricks. So take the time to visit those pages. Best practice guide is something that we generated last year in the context of a European project, Center of Excellence, BioXcel. So here they are all kind of do's and don'ts specific for different scenarios of that you can have with HADDOCK. So how to prepare structure. So we are quite picky about the input structures because we want to avoid problem downstream during the computing, how to define information about interactions, how to analyze the results. And you see here also an overview. So this is part of this best practice guide, but you see an overview of the different type of system that HADDOCK is supporting. So we support small ligands, we support glycans, peptides, nucleotides, proteins, and at a coarse grain level. And each type of molecule comes with some specific advices, tricks, do's and don'ts. So if you are going to do docking in one of those type of system, take the time to look at those in the best practice guide. We also have a lot of online tutorials that you can follow to get to know the software. Most of those are meant to be used with the web portal. We are running quite often workshops online last year, but also in real life. And we like to use the web portal because it's easy for most users. Even if you don't have experience with Linux, for example, you can run a lot of those tutorials. If you want to run the local version of HADDOCK, this one, the HADDOCK 2.4 local installation tutorial, you might not need to install the software if you were to use SBGrid one, but you should look at the different steps to set up the docking. I mentioned that HADDOCK is being developed, among others, under this BioXcel Center of Excellence, which is a European project, which is meant to further develop software and bring it also toward exascale. Another well-known software under BioXcel is GROMACS, for example, for molecular dynamics. And under BioXcel, we are actually running our support forum. So if you go to ask, excuse me. If you go to ask.bioxcel.eu, you will find different support forums for different kind of software, one of them being HADDOCK, and you can ask questions there and get answers. But you can also search, very importantly, before asking a question, take the time to search, because the question might most likely will have already been asked and the solution already provided. And you see some of these are highly used.
Just to break in briefly, we lost your slides.
You lost my slide, okay.
And they're back.
So I was mentioning the support forum of HADDOCK. You might have heard it, but you have not seen it. So askbioexcel.eu, you can search it. If you want to ask questions, you should register, but otherwise it's free to search for answers. So before asking a question, go there and look if the answer is already provided. And you can see from these topics that some, like dealing with elemental ions, is something which is coming back quite often. So to do with that, small molecules, dealing with dimers. So there are different topics that are coming back and back many, many times. Now, if you're going to use HADDOCK in a local setting, or if you're going to run it using the SB grid information, the installation, you will have to do a number of things yourself. So you will need to prepare the input PDB files. So the input for HADDOCK local is still the old PDB format. So, you know, if you know about crystallography, you might have double occupancies, meaning that some side chain in your structure might have two conformations. So you have to deal with those before doing the docking, because you want to only have one conformation, otherwise things might go wrong. So you need to remove those duplications. If you do an ensemble docking, so you're providing an ensemble of conformations, you will have to split the ensemble, the PDB file, which might contain many different conformations, into single PDB files. The server will take the ensemble file in one go, but for local, you need to split those. You will have to worry about protonation state of histidines. Very important. Histidines have a pK of around 6.6 on average, so they might be charged or neutral. If they are neutral, they are two-way, so you can put a proton on the side chain. So these are things that you will have to worry about. We have, I mentioned several times already, the PDB tools, which allows you actually to do some of this checking of PDB files. Remove double occupancies, remove insertions in an antibody, for example, renumber chains, change seg IDs, change chain IDs, select chains from PDB. So have a look at this. This is really making your life easier if you are dealing with PDBs a lot. It's also available as a web portal, by the way. You will have to generate your restraints file to guide the modeling process. So you might want to manually define your residues, manually generate a restraints file in CNS syntax. You will have to define a parameter file that contains your input data. That's the first file required by HADDOCK to set up the run. And once you have called HADDOCK once with run.param, you have a directory structure which is created. And then you will need in that direct structure to edit a file that contains all the parameters for the docking. If you are dealing with small ligands, you will have to provide topology and parameters for your small ligands. And there are different options to do that. If you use the server, this is done for you automatically. In general, you know, if you're using a local version, don't start from scratch. HADDOCK comes with an examples directory with different scenarios. Some of these are NMR specific. You have example of coarse graining, a trimer, peptides, small molecule, nucleic acids, EM refinements. So look at those, try to copy the setup from those. Look also at which parameters are changing those particular examples to apply ideal parameters or optimal parameters to your specific problem. You also see a refine complex option here, so you can also refine and not only dock. And again, if you're going to run local version of HADDOCK, take a look at this HADDOCK 2.4 local tutorial version. It will guide you for the initial steps. If using the server does a lot of those steps for you. We validate the input PDB. We will check for duplication and point to those. We are not going to remove them for you. That's your responsibility, but we are doing those. We check that the ions are properly defined. So there's a problem in naming. We will check for multiple occupancies. So these are checks that are done, and there will be an error reported to the user if we find some of those, but we're not going to fix the error for you. We automatically define the protonation of state of histidine by running mod probability. You can override the choice made by the server. If you give a list of residues that you think are important for the binding, HADDOCK will generate automatically the restraints for that. You might have a system where you have gap in your sequence, and when you do your refinement at high temperature, the different fragments might drift apart from each other. So when we recognize gaps, we automatically define these restraints to keep the molecule together. We validate the input restraints files. So we do a syntax check. We do not check that the atoms are existing. So if you define restraints to atoms that do not exist, they are not going to be used, obviously. we automatically generate topology and parameters. Depending on the molecule that you select, you might have optimal settings for these particular docking scenarios, and the server will do a post-analysis of your docking results, which when you run locally, you will have to run yourself. But the manual explains you how to do that. So this is a view of the server. If you go to the submit page, you can change the number of molecules to go to larger number of larger systems, basically. You see the options that the server in terms of molecule is supporting, so protein or protein containing a ligand. You might want to dock only a ligand, peptide, glycans, nucleic acids, DNA or RNA. You might be defining a molecule which is a protein nucleic acid complex, like you have a nucleosome and you want to dock a protein onto the nucleosome. And you see this shape option that I just mentioned when I was speaking of this new shape-based protocol for protein small molecule docking. This was the regular submission interface. There is also, when you submit a file first, you can save a file, a JSON file, which contains all your docking parameters and all the input data, except the EM density files, which are too large. If you save this file, you can also edit manually this text file. And instead of going through all the forms that you have to change at the submit level, you can use this submit file option where you simply upload this one parameter file and the docking is large. So always basically save this JSON file. And at the end, I want to mention also a refinement option. So if you don't want to do docking, but you have a model of a complex you want to refine, maybe to remove clashes at the interface, or you have a low resolution EM model you want to refine, a complex, you can use the refinement, and we are currently 4 different protocols implemented. So here you will upload the PDB of your complex. HADDOCK will already distinguish what kind of molecule you have, and then you can proceed with the refinement. The server also tells you which type of modified amino acid are supported. This also applies to the local version. So the list of molecules that you will find here are also supported locally. And if you look here, you will see, for example, so these are all the current carbohydrates that we support. So the naming should be proper. You also see the ions that we are supporting and how you should name the ions if you want HADDOCK to deal with them properly. A number of cofactors, and here you have a long list, you only see 2, of modified amino acid. Okay, so if you want an acetylated lysine acetylated on the side chain, you should change the residue name to ALY. You don't need to add the missing atoms because HADDOCK will build those missing atoms. This is true when you use the local version and when you use the server. With that, I want to close to leave some time for questions. But before doing that, I should also thank all the people who are contributing to our constant development of HADDOCK. And this is a picture of my group earlier this year. Of course, all online. This is how we've been holding our group meetings since a long time. Funding agencies, NWO, local Dutch Research Council, and European grants over the years. I also want to mention here again the Open Science Grid, which is supporting us by providing computational resource, and SB Grid also for their support over the years. All the previous developer of HADDOCK, so this was a meeting that we had in 2017, where we have been trying to do an optimization problem. This is a hard problem because it's a very flat surface with a very small minimum, not easy to find. And finally, thank you all for your attention. And I will be glad to answer any questions.
Any questions? Go ahead and questions, you can put them in the chat if you want to send them. You can also click the raise hand if you have a question and I can call on you. You can ask your question directly. So any questions? I mean, I have a couple. So just on a practical note, is it always a 2-component docking, or if you had a an interface that sort of if you had 3 subunits or something, is it possible to do this?
Yes, so I showed you a slide of the server with the submission interface. So there you define the number of subunits that you have, and the forms will be automatically adapted to the number of subunits. In the examples directory, which is provided with the local installation of HADDOCK, we have an example of a trimer and of a tetramer, I think. The current limit is 20. The machinery is built to go up to 20. So you can also use that to cut a protein into pieces, for example, if you have domain motions, this kind of thing. This is an approach that we have used in the past, but the more subunits you have in your docking process, the more challenging it becomes to get to the right solution, because of course the number of degrees of freedom that you have to sample increases exponentially, very quickly.
And does that run into like a sort of computational limit? Like it's just you wait too long, or is there a?
It's yeah. So you have, well, it all depends on the data that you have at hand. If you have good data, you can also simplify that by going to coarse graining, for example, so that you reduce the number of particles. But it's it's a molecular dynamic type of problems where you have maybe n log n scaling. And if you are doing things really badly, you have n square scaling. So the larger the system, the longer it will become. And also the energy minimization at the beginning, we increase the number of steps as a function of the number of molecules. So this becomes more heavy in terms of computations. And you might say, if you go from a few seconds for a dimer to maybe half an hour for 10 MERS, it's not that much, but if you have to do 10,000 times half an hour, it starts becoming a lot of computing. So right. You need, if you want to run it, you need to have, say, ideally, I would say you can probably run it on one node which has 48 cores, or even if you have probably a laptop with 8 or 16 cores, you're going to be able to run things in a reasonable time. But if you have 100 cores, then it's going to be way better.
Mm-hmm. Sort of a related question that came in over chat is how does HADDOCK handle like water or ion mediated interactions in an interface? Say you have a water mediated interface, can you specify that, or maybe an ion?
Yeah, so we have in principle you could even give a water molecule as part of your PDB, and it will be kept. And this is also dangerous, because if you were to download the file directly from the PDB and give that straight into HADDOCK, you're going to end up with water molecules. You're going to end up with crystallographic junk, meaning part of the buffer which is there. So look always at what you have in your PDB. We also have developed an option. This is already quite some time ago, where we do what we call solvated docking, exactly for the cases where you expect that you have a wet interface. And there are examples of that. And for nucleic acids, probably it's even more relevant. And what's happening is that you basically add one layer of water to your molecule before docking, and then dock with this layer of water. And you will have to get rid of that water because you might not need that water. So there's a Monte Carlo process implemented, which will randomly remove those water, but based on probabilities of finding water-mediated contact. So we have two scales. One was derived from an analysis of crystal structure. So where, for which combination of amino acid do you see more often water-mediated contact? And another scale was derived on the Kite-Doolittle hydrophobicity scale of amino acid. So these options are there. In the examples directory of HADDOCK, the local installation, there is a solvated docking example that show you how to do that. On the server, you flip a switch and it's happening. In our hands, it only makes sense to use that first if you really suspect that it's going to play an important role, and if you have good data. If you have very lousy data and no data, don't waste time, because this adds computational time to the search. So it doesn't, personally, in a group, we almost never use it. Okay, the option is still there, but I think there are users using it, and users telling me that it's helping them a lot for their particular problem. So it's there, but it adds computational time. So you use it wisely. And if you don't know what wise is, ask us, okay, or use the forum, but ask before doing crazy things.
And I guess on that note, are there restrictions in place on the server side to keep people from firing off jobs that would consume an inordinate amount of resources?
Yeah, so we have so on the server we put limit on the maximum number of atoms. We also flag if people define too many restraints. So we had people, so we are constantly trying to solve issues on the server, because people are very creative in what they can do or what they will do actually. And then there are things that you never thought that someone will use or a combination of options, and people do it, and then you run into problem. So we are building all kind of safeguard mechanisms on the server to deal with that. If you're running locally, then it's your problem. If you're using all your cluster, it's not our problem. When you use the server, you have to check for that. The other thing is that we have a maximum number of active docking runs per user. So I think the current is 5 or 7, so that we want to avoid the problem where one user submits 1,000 docking runs, and this happens actually. Okay, so some users have a way of filling the forms automatically, probably, or if you deal with the JSON form, it's probably much more efficient to do that. But that will not create a queue of 1,000 jobs before the next user can go in. So you have to have this fair share mechanism, and that's the machinery in the back of the server, which has been refined over many years, basically. And from time to time, we do cancel jobs because we realize that people have been doing something silly. So there's this job which has been lost there on the server for a long time. Nothing is happening. So if we detect those, so it means that someone from time to time has to, go at the terminal level and do some checks and see what's happening. And if this happens, then we usually cancel those runs, notify the user friendly. And if they keep doing it, we might get a bit more aggressive in our mailing. But so you have to educate your users also. So we're trying to make their life easy, but it's sometimes you have to be careful what you're doing. Usually users are also very good at pointing problems to us, because they say, I submit my jobs, but it has not been running for 3 days. It means that something might be happening, and then you go check. So it needs a bit of babysitting.
Yeah, I'm familiar that users are very good at pointing out all of the problems with the software environment. So,
but sometimes they do
things,
and sometimes they do things that you didn't expect to do, but which are cool things, and you realized, we can do that. Amazing, and it works. So that's also great. The other part of it.
Yeah, that's fantastic when that happens. So I saw Pete had a question. Pete, jump in.
I had a couple, and I think one of them you folks mostly answered already. So the one that's left is kind of possibly a stupid question, but when you were talking about ambiguous restraints, delete 50% for each run, would there be any advantage to putting in a restraint with a probability score associated with it? Like, I'm 75% confident with this restraint, I'm 25% confident with this other one. Is that like, could you say anything about the idea about using some of those kinds of approaches?
Yeah, you would have to go deeper into that. Then you would have to go into CNS, explore, and code for that. So you have to change the backend machinery for that. They are also, because if you might know IMP, where you might have in IMP, you might say that, but it means in IMP they are not using the restraints it's a Monte Carlo process, so you don't calculate forces. And the forces are distributed onto the atoms, but you might just, discard some distance when you do your Monte Carlo, your check, your energy evaluation. And then you might discard the distance with a given probability. So that's a simpler things to do. So if we were to associate these probabilities, you will have to weight the forces. So you need to add something to your entire machinery so that those weights go down. And what we cannot do currently, you can define different force constants for different classes of distances. So I define there are 3 ways of input it, but you cannot define a force constant per restraint. So the machinery is not there.
Yeah. Okay. I don't have a particular use case for it, but it's something that it definitely popped into my head.
It will be interesting to do that.
Jonathan Dorval had an interesting question. If you want to unmute, you could ask your question yourself, or I can pass it along. I can go ahead and ask it for him. The question was, can we use a SAX envelope as a template to position the proteins or domains inside the envelope? Sort of using the shape function to be able to restrain based on,
Yeah, so that's something that we are writing that story right now. So basically where we use the SAX, and then you can use, you generate a bid model from your SAX data. And then I show you this way of using the shape for the small ligand docking, but you can do the same with your proteins. Where then, so the idea is to have this shape, these beads that are defined. And then you could define, for example, a distance restraint from your C alpha carbon to any beads. So then you have a very high ambiguity, but you're basically pulling your molecule. They say you should overlap with this shape. And next to that, you do your docking. So that's something that's, so the shape input in the server is for that purpose, actually, and also for small molecules. So we're still using distance restraints to pull the molecule inside the shape.
Yeah, that could be
it will be on
very interesting.
bioarchive later this year.
Yeah, I can think of other cases where it might not necessarily be, a SAX envelope. It could be a low resolution cryo-EM map, right? It could be,
maybe it's more efficient. So we'll have, because the EM implementation is quite computationally heavy. So maybe, if you have low resolution map, you are better off defining a few beads to represent your density and then using this shape type of restraining rather than going for the density evaluation. So that's something. So in principle, it's very versatile. You could use it for any type of volume information.
One other question that came in was any machine learning inspired updates in the pipeline for HADDOCK patterns of dihedrals found at the interface, etc.
So not so much in the energy functions. I guess we will have to switch to another software want to do that. But moving away from CNS Explore is very hard because of the incredible flexible syntax. For those of you who know the code, and most of pretty much all the docking is in HADDOCK is coded at the script level, okay, in CNS. So that's and it's hard, and the selection syntax is so powerful. So what we have been working on is more on the scoring side. And so we have been doing deep learning. So we have a deep learning project, Deep Rank. It's actually on bioarchive, so you can find it there. And there's a GitHub repository where the idea is to basically transform the interface of your complex, of your model, into a 3D image. So you put a grid on the interface, you describe different features there, which can be position of atoms, but charge, conservation, and you generate 3D image of your interface. And then you can train a model to distinguish native from non-native. And you can also do the same instead of using these 3D images, which is quite computationally demanding, and which also depends on the orientation at which you look at your complex by going to a graph representation of the interface. So these are things that we have been, that we are still working on. We have also published some scoring function, graph-based scoring functions called iScore, where so the machine learning aspects at this time are more for the scoring part than not generative models in a sense.
Yeah, that makes sense. I mean, I think one question that I had is that, well, CNS, CNS and Explorer have been around a long time. So, but, they're flexible and they're very fast, right? So, this is proven technology. So no plans to, yeah, exactly. No plans to move away from, from using that as the back end anytime in the future.
So within the BioExcel project, we have always been talking of could we use, for example, GROMACS. I don't know if some of you use GROMACS. Try to define an ambiguous restraints with 1,000 different atoms in GROMACS. That will be a nightmare. So you lose this flexibility.
Yeah.
So we need a software which allows us basically to have this flexibility. During the refinement stage of HADDOCK, when we define flexibility, you basically define a selection syntax that says the side chains of any residue of chain A, which is within 5 angstrom of any residue of chain B, is going to be flexible. And you almost write the command as I'm speaking it out. And you can update this flexibility definition during the run. You don't need to exit. If you were to do that in GROMACS, you will have to generate a new TPR files every time you change something in your topology. So you go out, you go in, you go out. Another option could be OpenMM, which has a Python interface. By the way, GROMACS also has a Python interface now. I think that would be nice to interact at the Python level. So if we can start doing atom selections, which are key to a lot of what we are doing at the Python level, and then give that to the GROMACS engine or to the OpenMM engine, maybe in the future, there will be a way to move away from that. And actually what we are doing now is we are working on HADDOCK 3.0, where we are breaking apart things into small modules so that you will have flexibility to build your whole pipeline and also incorporate third-party software. So maybe along those lines in HADDOCK 3, there will be links to OpenMM or GROMACS for some of the part. You might still need some old part for maybe the rigid body docking, and then you could switch to something else. So we are thinking about it, but it's not simple.
Yeah, it's definitely, it's hard to switch out the foundational tools, right? It's
yeah,
that's pretty challenging. I guess last question, and we can wrap up. Are you seeing more cryo-EM users as EM sort of comes online? And Ken asked, are you seeing more cryo-EM users than NMR as EM gets hot?
We don't monitor what people are doing, so I would not know. I know that they are EM. I don't think we have more EM users than NMR people. I think the people who are doing, EM, they don't need docking probably because they go for the high resolution stuff. So, but they are still, what we see happening quite a lot actually is they are still, we see people who are doing EM. We might not have the full resolution that they need to completely build complex, and then they come to HADDOCK to refine the interface of the models that they have built. So HADDOCK being used as the final stage basically to refine those interfaces. And actually, we have put also a paper in bioarchive on that topic what is an optimal protocol.
The glue for hybrid methods, right? Like you can take, you've got some mutagenesis data, you've got a little bit of cryo-EM data, resolution, you can start to put the whole picture together, right? So,
and you have IMP, which is also a software used very much for that. I think IMP can have representations that are even more coarse-grained. So we are the coarse-grained on atomistic level, and IMP can go much higher to even deal with molecules for which you have no structure, where you just put a sphere out there. So, so it's a different scale. So we are much more focused on atomistic to say coarse-grained, and IMP is going higher up in the scale of integrative modeling.
That's interesting. Well, with that, we can wrap it up. Alexandre, thank you very much. It's always great to hear what's going on with HADDOCK. I'm interested to see if your spike in COVID usage turns into a spike in citations in about 6 months. That would be really
we'll
cool.
see. I think last year there were quite a lot of citation as well. We'll see.
Yeah. I mean, you've already got a lot, but still, it would be cool to see a spike there. So great, and thank you everyone for joining. Have a great summer. We will see you in the fall.